Hello everyone and welcome to Cure Talks. I'm Priya Menon and today we are discussing CAR T-cell therapies and multiple myeloma with Dr. Shaji Kumar, Professor of Medicine at Mayo Clinic. Mayo has a large CAR T-cell program and Dr. Kumar is one of the doctors working with a focus on myeloma. Joining us is a myeloma panel of patient experts, Gary Petersen, Jack Aiello, and Cynthia Chmielewski. Thank you for being here. We will be taking in a few questions at the end of the discussion based on the availability of time. So please add your questions to the comment section of the page, or you can also email them to me, priya@trilix.com. Getting started with the discussion, Dr. Kumar, thank you for being here today. I want to start with something which is on everybody's mind, and I would like to address it first before we dive into the CAR T therapies. That is COVID-19 and the vaccine rollout. Would be great if you could comment on the COVID vaccines and what you are telling your patients.
Yeah, thanks, Priya. I think, obviously that's on the top of everyone's mind, right? They think how to kind of stay out of the way of that infection until everything comes, gets back to normal. So I think the good news is, we have vaccines, and not so good news is we just don't have enough of it yet. But hopefully that will change in the near future. So at least in the US practice, currently we have two vaccines that are approved, both the Moderna vaccine and the Pfizer biotech vaccine, and both of them appear to be quite comparable in terms of their efficacy. The Pfizer vaccine needs a little bit more colder storage facilities, and is given 21 days apart. The Moderna vaccine is given 28 days apart and appears to be maybe a little bit more stable at higher temperatures. Now, both of them have the same technology behind it, which has not been widely employed for creating vaccines in the past, but clearly it's a technology that is quite fascinating and has become now with these vaccines mainstream, and hopefully will allow us to create vaccines in the future too. So the big questions that come up is, first of all, should myeloma patients get the vaccine? And the simple answer is yes, everybody should get the vaccine, including the myeloma patients particularly. Now, the second question that always comes up is it going to be as effective in the myeloma patients as it is for normal people, normal individuals? And the answer is we don't really have good data on that. Nobody has done a systematically a study looking at the immune responses following vaccination. I suspect we will see more of those coming out in the next few months. Until we have more definitive data, we should just work on the assumption that at the minimum it will be better than not getting a vaccine. Even if it isn't a 95% effective as it is in the normal individuals, it still is going to be valuable in one, potentially preventing the infection, and even if it doesn't prevent the infection, can decrease the severity of infections when somebody gets if someone gets it. So so then the question is, when should someone take it, right? Especially in the context of the different treatments we use for myeloma. So if someone has, let's say, smoldering myeloma or active myeloma who is not on any treatment, there's no difference than anyone else. If somebody just had a stem cell transplant or had a cellular therapy, the recommendations widely has been give 3 months from the chemotherapy associated with that particular therapy. So again, conditioning therapy for the transplant or the lymphodepletion therapy that was given prior to the CAR T-cell therapy. So wait for 3 months. Now, how hard and fast is the 3 months? It's not, there's no data to say it has to be 3 months, right? We are just basing that on our experience post-transplant, looking at the history of the risk of infections in general. So if someone, I think if someone has the opportunity to get a vaccine, let's say 60 days from the transplant, I would be okay with them getting it, knowing that they're going to get a booster dose 30 days, 28 to 30 days after that. So, and now what about people who are getting other treatments, right? If somebody's on, let's say, lenalidomide maintenance, I think should be just fine. Now, the big question we don't know very well would be the people who are getting either cytotoxic chemotherapy or people who are on the monoclonal antibodies, both of which we know can depress your immune system. And in those settings, in the absence of clear data, I think what we should just strive for is, when you get access to the vaccine, take it. because, given the scarcity, we don't want to pass upon that opportunity to get vaccine because of delays. If you're able to get, few days before and after the vaccine without getting any infusions, I think it would be great. But I wouldn't dramatically change the schedule based on the vaccination dose itself. Because, as we have seen from all the data we have, the biggest risk for the patient is myeloma that is not under control. So I don't think we should compromise on the myeloma care to facilitate the vaccine. So, again, nothing is ironclad. Everything has to be in the context of the individual patients. Now, finally, I think there are other vaccines that are going to come online. The Janssen vaccine looks like the data is good. So we will get a better sense in the next few weeks. The advantage of that is it's more stable, easier to transport. It's a single dose. So that's a huge advantage. Then outside of the US, the AstraZeneca vaccine is also approved and is being used across the world. It again is 2 dose, but much more easy to transport and so forth, and does not use the mRNA technology. And I think, so there are opportunities or options, but definitely the key thing is get vaccinated.
Thank you. Thank you very much for answering that, Dr. Kumar, because we've had quite a few queries from myeloma patients as to when and whether they should take the vaccine. And here our panel, Gary has, I believe, been vaccinated. Jack and Cindy are still for vaccination. So yes, thanks for answering that. And that kind of look forward to for all the patients, myeloma patients. Yeah. So, Dr. Kumar, my next question is going to be kind of one that will just give us a little summary of the ASH 2020 meeting. I know there were so many new and important developments there. Many of them on CAR T therapies, which is the focus of our discussion today, and which the panel will be diving in deeply for discussions later. So, could you provide some insights on what you feel the highlights and the major takeaways were from the conference, which are non-CAR T?
Absolutely. Priya, I think this was, this ASH had lots of information in their data, even though none of those necessarily might change how I treat my patients today, but it really gave a very nice glimpse of what's on the horizon, how the myeloma field is going to change over the next 5 to 10 years. And I think it's going to be dramatic based on what we have seen, particularly with respect to immunotherapy, and I know we'll get into the CAR T later on. So, in terms of thinking about the new developments, I think you have to put it in the context of how we deal with this entire spectrum of disorders, right? So on one end, we have the concept of early intervention for the precursor stages. So there's increasing numbers of trials that are being done in the smoldering multiple myeloma phase. And I think, we have the advantage of having a precursor phase to this disease that lasts for 10 to 15 years at the minimum, and a test that can detect that, which is very simple using a blood test. So we have a good opportunity to identify people with these conditions ahead of the time when they get that myeloma. The second big question is, what can we do about that with that knowledge? And I think clearly the trials have shown that at least in the people with the highest risk of progressing to myeloma, some kind of early intervention appears to have some benefit. Now, we don't, where there's still a lot of clarity that needs to be gained in terms of, how can we be absolutely sure that we select the right patient to minimize the risk for the others? What is the best approach? Is it a treatment like myeloma or a less intense treatment? Or maybe it is a different for different patients depending upon certain characteristics that we need to identify. So while there was not a lot of data in terms of actual regimens for treating smoldering myeloma, there were a lot of abstracts that I looked at the biology of the disease, which will help us refine the whole risk stratification system. So we have the new 20-20 system for smoldering risk stratification developed by Mayo data, and then subsequently validated using the International Myeloma Working Group data. Now the question is, how do we build on that foundation? And I think genomics, the immune profiling, looking at the epigenetic changes, all those will contribute to making that a more specific system so that we don't overtreat anybody, but at the same time, we don't leave somebody untreated who really would have benefited from treatment. So I think the biology of smoldering myeloma with the new technology that's becoming available is going to get really dissected over the next few years, and we will have a better sense of what that's going to be. The next step is, looking at the whole concept of risk stratifying multiple myeloma and individualizing therapy. Now, obviously, that is an area, again, rapidly evolving. We always have the traditional fish-based high-risk stratification, which obviously is the most important. But on top of that, again, some of these new mutation profiling and the immune profiling will add additional layers of beneficial information to that risk stratification. Then moving from the risk stratification, treatment of newly diagnosed myeloma. So we saw the updated data from the Maya trial using the daratumumab Lendex. We know that the median, even though we still haven't reached the median of progression-free survival, it's reasonable to assume that's going to be around 5 years with that triplet. That to me is a big jump ahead because 5-year PFS, we have always thought about in the context of a transplant in a transplant eligible patient population. We have not quite reached those numbers in the non-transplant eligible patient population so far. Obviously, we don't want to compare regimens, but this is extremely promising for the non-transplant eligible patient population. And the questions going forward is, how do we build upon that regimen? Do we build upon it by adding more drugs, replacing something in that triplet with something else that's new? Now, in the transplant eligible patient population, I think the big question now is, do we use 3 drugs? Do we use 4 drugs? Obviously, we saw the updated data from the Griffin trial showing that you can get deeper responses, more patients who are MRD negative prior to stem cell transplant. But the data is still pretty early on to say that we should be giving everybody 4 drugs because there's a price to be paid both in terms of toxicity and the cost of therapy. So I think as we get more data from those studies as well as other larger studies, we will be able to identify who, if any, benefits from using a 4 versus 3, or are we better off using that drug in a subsequent treatment later on after transplant. Now, talking about transplant, there's clearly at least 3 sets of data which suggested that transplant still plays a role in those patients who are eligible to go through a transplant. We saw the updated data from the French IFM 2009 trial, which showed that even with a 90-month follow-up, the PFS is still about 14 months better with transplant versus no transplant. But it also told us that the transplant did not improve the overall survival as long as these patients accessed transplants at the time of relapse. So I think that early versus delayed transplant strategy still, looks like they are equivalent, providing patients with a choice to delay transplant if that's the way they would like to approach this. And finally, we also saw the updated data from the EMNO2 trial, again showing that transplant is better than a non-transplant consolidation. Now, that also has to be taken in the context of the data with tandem transplant for high-risk patients, right? So there is multiple trials which have suggested that maybe there's a benefit for the high-risk patients to get a tandem transplant. I know the jury is still out on it and people have very strong beliefs one way or other, but it is something to be kept on the table when we deal with someone with high-risk myeloma, because we have not yet made substantial progress in those group of patients. And there's clearly work that needs to be done for the high-risk myeloma patients. Now, I think the post-transplant maintenance or consolidation, I think it's, the maintenance is fairly straightforward. I think lenalidomide has become the de facto standard based on the clinical trials. We know that the high-risk patients would benefit from getting a proteasome inhibitor plus immunomodulatory drug. Now, where does daratumumab or any of these newer immunotherapies stand with respect to maintenance? We still don't have good answers for that. Now, kind of moving on to the next part, right? Unfortunately, we still end up having to deal with relapsed myeloma. So what do we do for patients with relapsed disease? Obviously, the first relapse is now later and later. Previously with the previous therapies, we used to see the first relapse on an average of 2 to 3 years. Now we are seeing the first relapse on the average of 4 to 5 years from starting the initial therapy. But that first relapse often happens in the context of 2 different things that are different than what it was 10 years ago. One, patients are often on maintenance therapy, right? So they are on low doses, often low doses of lenalidomide. The second is patients come into that first relapse in a much, much better shape than what they used to 10 years ago when the treatments were a lot more toxic and the treatments were not that effective. So in a way, the first relapse patients that we see in 2021 is almost like the newly diagnosed myeloma patients from 2010 in terms of their overall well-being, their functional status, and the opportunities that are available for treating or controlling that disease. So what do we do for the patients with myeloma, newly diagnosed, who are often refractory to lenalidomide? So we need to find regimens that are free of the lenalidomide containing combinations. The good thing is we have lots of different options. So we have the daratumumab or isatuximab with pomalidomide. We have daratumumab or isatuximab with carfilzomib. And we also saw the carfilzomib with cyclophosphamide, a combination that was presented by the Spanish group. So we have 5 different regimens we can use in these patients. And obviously there's a lot of other drugs that are coming up too in terms of the coming up the ladder. But at least in that first relapse in the lenalidomide refractory patients or the Revlimid refractory patients, we do have good choices of triplets that can control the disease for, again, another 2 to 3 years, which again is a substantial improvement from before. And this is going to be an important question because, we are going to see some of these patients getting treatment for smoldering, also coming in with their first progression, so to speak, who are refractory to lenalidomide. So what we learn in this setting is going to be something that could be translated to those patients as well. So it's an important fund of knowledge that's going to be helpful. Now, beyond that first relapse, second relapse onwards, now we have a whole lot more choices of drugs. We have the newer drugs like selinexor and belantamab, both of which have been approved. We have data from combinations of selinexor with carfilzomib, with pomalidomide, with belantamab, with proteasome inhibitors and immunomodulatory drugs. So we have the opportunity to use many of these newer drugs in various combinations. Again, the underlying principle being, can we come up with 3-drug combination, which includes drugs that patients have never seen or only have seen some earlier version, and definitely not refractory to that particular class of drug. So clearly, those are all options that we have for the third and the fourth relapse. But I think what is the most fascinating, obviously, is what's coming through, right? Which is the immunotherapy. And there again, I think, for the longest time, we thought the immune system in patients with advanced myeloma just didn't have the ability to react to the tumor cells. Now we have data, whether we're talking about CAR T cells or whether we're talking about bispecifics, there's no doubt the T cells that these patients have are capable of getting rid of the myeloma, but they need some additional help. And how do we provide that? The 2 approaches that I think, one is obviously using the CAR T cell approach we'll talk about, but the bispecifics. I think the bispecifics, we saw a bunch of them being presented, majority of them targeted towards BCMA, which is the same target as the CAR T cell and the drugs like belantamab. But we also saw 2 bispecifics with different targets. The one targeting the FcRH5 and also the GPRC5D. Both of them appears to be equally effective as the BCMA targeting bispecifics are. So I think we are going to have a lot of different options. I think the key would be figuring out where do these different immunotherapies fit in, both by itself and in combination with the treatments we currently use.
Thank you, Dr. Kumar. That's very comprehensive. Thanks a lot. With that, Gary, please start with the discussion.
Yes, thank you very much. And thank you for that excellent introduction, Dr. Kumar. Now, one thing I found over the last several months is that all new classes of drugs have been approved in the last 16 years have had dual designations, meaning that it's an orphan drug designation by the FDA with another one like fast track breakthrough, etc. They have been given a special preferred status by the FDA. We now have 7 such dual designations for myeloma, and 4 of them happen to be CAR T. So that it's taken center stage with regard to myeloma treatment. Now, I was there at the presentation that you gave on CT053, and I thought that was pretty impressive. And I figure that you must be very well versed on CAR T. Now, first thing is could you explain to our audience, a lot of which are newly diagnosed and not necessarily nearly as skilled as you are in understanding this disease, that you could put it in a patient-friendly format. What is CAR T? I know my first thought was that it was the first car made by Ford Motor Company, but that can't be true. So, because it has to do with blood. So I figure maybe you could provide me with a little bit more information with regard to CAR T.
Absolutely. No, I think it's a good analogy, actually, if you think about, Model T is what brought the cars to the masses, right? And hopefully, and hopefully these CAR Ts will actually get this treatment approach to all patients with myeloma at some point. So yeah, so let me start from what exactly is a CAR T, right? So generally, when you think about T cells, whenever they engage or whenever they encounter a tumor cell, it will again the T cell receptors that are naturally developed, or I should say resized or reformed on those T cells, are targeted towards the tumor antigens. Now, obviously, in the myeloma setting, we are not getting that natural response against the tumor cells, and that's why these T cells are unable to get rid of the myeloma cells. So what the CAR T does is it basically takes a T cell receptor and modify it in such a fashion that it now can target a specific antigen or protein that is on the myeloma cell. Now, the vast majority of the CAR T cells that have been developed so far has been targeted towards BCMA in the setting of myeloma, but other antigens like CD19 in the setting of lymphoma, for example. So whatever antigen you think is unique to a particular tumor can be used as your target antigen, and your T cell receptor can be made to target or attract that particular antigen. Now, the next step is to get those modified receptor into the T cells of the patient. So what we end up doing is collecting the autologous T cells of patients' own T cells using apheresis. You collect the T cells, you transfect them using a lentiviral vector. And what that virus does is it allows these the gene or the genomic region that codes for these modified T cell receptor and integrates that into the cell's DNA. So it essentially becomes a part of the cell's genetic makeup. And that is a permanent change that is brought into that T cell. And once that is integrated into the T cell genome, then the T cells will start making that protein, and the protein gets expressed on the T cells. And now the T cells are, targeted. Now it's like a, so it's basically the only antigen it will recognize and get stimulated by would be the BCMA or whatever antigen that we are targeting against. And this is different because there are a variety of ways you can introduce genetic material into a different cell. A lot of them are transient, so what we call the transient transfections, which does not allow the genetic material to get integrated with the cell's genome. Those ones tend to be transient. They are in the cell, they make a bunch of protein, and then that genetic material gets completely wiped out. Here, what we want is a permanent change in the T cell. So that's why we use the lentiviral vector. Now, the disadvantage of using a lentiviral vector is theoretically, there could be random insertions of this genetic material, which can lead to some mutations, or theoretically, it can cause other problems by modifying the genome in unintended fashion. As a result, that is why many of you have noticed that these clinical trials will have you sign you up for up to 15 years of being followed up, because that is something the FDA wants to make sure there are no long-term impact of that. Now, we will talk about it later, but there are other technologies that can potentially go around this problem. Now, once we have these T cells, the autologous T cells transfected with the lentiviral vector, then they make sure they are all expressing the particular T cell receptor, and there are minimum threshold in terms of the percentage of cells that should have it. And then that product is then reinfused into the patients. Now, the problem is if we just reinfuse it, there's a high probability that it can get destroyed by or it just may not be able to do its job if there's a completely intact immune system that's already there. So we use that lymphodepletion chemotherapy ahead of time to get rid of the lymphocytes, or at least decrease the lymphocytes that are already there. And then this autologous CAR T cell then goes in, it recognizes the tumor cells, and it creates what is called as a immunological synapse, which is basically a connection or a tunnel between the T cells and the tumor cells. And then they basically inject a whole bunch of other kind of chemicals which lead to the cell death. And so it's basically, it's highly targeted, very specific for the tumor cell. It will not care about whatever other cells are around, and just specifically kills the tumor cells. Now, will it kill all the tumor cells? We don't think it does, because of the fact that the majority of the patients still have relapse at some point in time. So obviously the ongoing work is trying to see how can we enhance the efficiency of that, or how can we figure out why some tumor cells escape this. Now, the other part about the CAR T cell, I'm sure all of you heard about, is the kind of toxicity that we see with them. And that is related to all the chemicals that are released as part of this tumor cell kill, because the same chemicals that these T cells are secreting to kind of enhance the immune reaction and kill the myeloma cells also lead to kind of a systemic reaction to those, which result in the cytokine release syndrome that is often seen in these patients.
Well, thank you very much. The other thing I would like you to discuss is really, and not so much about ide-cel and cilta-cel, or BB2121 and J&J 4528. Those are going to be covered in depth by Cindy and Jack, so I don't want to steal any of their thunder. But you gave a presentation on CT053, and you were also there when they also talked about BCMA 101. And they seem to have some special characteristics with regard, I think, to speed of development of the CAR T. You know, they were quite a bit faster, maybe, and also seem to have a longer overall survival and a few other things. So I was wondering, you know, if you could talk about those things, but prior to that, just tell us why there's so much enthusiasm around CAR T to begin with, you know, given, you know, cytokine release syndrome and all the things that, all the hurdles that exist.
No, absolutely, Gary. I think the reason why we are all enthusiastic about the CAR T cell for two reasons. One, you know, it is proof of principle that you can use your own immune system to actually get rid of the myeloma, and that is quite, you know, that's a paradigm change to me, because, you know, we were worried that it may not be able to do that. Second, obviously, the responses that we have seen, right? So we have seen patients who have seen, you know, 14, 15 lines of therapy, and they get to be a minimal residual disease negative, right? I mean, we are all quite stunned by some of the images that have been shown of a PET CT that is, you know, completely, you know, vividly colorful, and then a month later has no evidence that there's myeloma. And we have, as with this particular CAR T as well as others, we have seen that, you know, after you infuse the CAR T cell, 4 weeks later, you don't see any myeloma cells in the bone marrow. You go from being packed to completely not there. So I think those are dramatic things that we haven't even seen with some of the treatments that we consider standard for today's treatment. So clearly, you know, we cannot compare any of this with anything within the absence of randomized phase 3 trials. But individually, based on what we would have expected, how these patients would have been expected to do, we are clearly seeing they're doing much better than what we have historically seen. So there's no question, these are all highly effective therapies. And I think the big question is, you know, who gets it, when do they get it, and what are the toxicities, and how do we manage the toxicities, and are there things that we need to be thinking about long term, particularly with respect to toxicities. So with that in the kind of the background, you know, I just want, maybe I'll jump into the CT053 part of it. Now, obviously, as you have seen, there's a whole host of different CAR T cells that are being developed. You know, there's obviously the concern that where are we going to fit all these in, and does one work and then the others don't work after that? I mean, to me, the more the better, because, you know, it's just like what we talked about with the vaccines for the COVID, right? I mean, the more vaccines, the better. Here also the same thing, the more number of options you have, I think the better we are. So with the CT053, you know, with each of these CAR T cells, there have been minor tweaks. Most of them have been done with the aim of one, either make it more tightly binding to the antigen, or it actually is able to stimulate the T cell efficacy is enhanced by changing the intracellular domains, or we create CAR T cells which are less differentiated in terms of their immune naivety, so that these actually may be more flexible CAR T cells, so to speak. And then finally, can we actually make these CAR T cells faster than the 2 to 3 weeks that we have right now? Because many of these patients cannot afford to wait for 4 weeks without treatment, or without effective treatment. So the CT-053 is, again, overall it's very similar, but it certainly seems is able, the manufacturing time appears to be shorter compared to what's with some of those first generation, I should say, the Model T CAR T cells, right? So now we are moving on to some of the improved versions of the same thing. So with the CT-053, it's again a dose escalation study. We enrolled patients on 2 different dose levels. 94% of the patients did respond to the treatment. Many of these patients were able to get to be MRD negative. Almost all the patients, we saw the bone marrow plasma cell percentage coming down to 1 to less than 5% in almost all the patients by the end of 4 weeks. When you look at the M spike or look at the serum free light chains, dramatic decrease, pretty much normalizing over the first couple cycles. So clearly it's effective. It seems to be working fairly rapidly, consistent with what we have seen with the other CAR T cells. And in terms of toxicity, it seems to be fairly comparable. We didn't really see a lot of cytokine release syndrome. Most of what we saw, and again, it's small, steady, so everything has to be taken carefully from that context. There were only like 20 patients, but the grade 3 or more CRS was not seen in any of these patients. There was only 1 patient who had a grade 3 neurological toxicity. So, and all these cytokine release syndrome was seen in the first couple of days, lasted for an average of about 4 days. About a third of the patients were treated with tocilizumab, and all the cytokine release syndrome was, very well controlled with the supportive care management that we have. Which also, I think, highlights another aspect about CAR T is that, we talked about why we are so excited, right? In the beginning, we were a little bit worried about these toxicities, but as we get more comfortable with them, and we know we can detect them early, treat them early with drugs like tocilizumab, we don't feel as worried about this. So clearly, we are more comfortable with toxicity and more impressed with the efficacy, and that's why I think the CAR Ts are continuing to move, kind of to the front of the pack, so to speak. Now, the responses, the follow-up is relatively short. I mean, we have patients followed up until less than a year in this particular study, so it's hard to know. But there was another study that was presented by the group from China who has up to 2-year follow-up with the same product, and they were able to show that many of these responses were pretty durable. They don't have the median PFS in their study, again, relatively small number of patients, was about 18 months. And the median duration of response was about 22 months. Now, granted, those patients were not as heavily pre-treated as the ones we put on this study, but it still tells us that you're able to get durable responses with this particular CAR T cell. So I think it looks exciting. This trial is moving ahead with a larger phase 2 with a regulatory intent, and at some point in time, also a phase 3 trial, just like what is being done for CAR T cells. And Gary, I think you wanted me to talk about the PBCMA one, but I'll just stop here to see if there are any questions before I move on to that one. Shall I jump on to the
yes, please.
Yeah. Yes. Okay.
So, we, like I talked about before, right? So using the lentiviral vector has some disadvantages of, again, potential disadvantage of genetic changes. So what the PBCMA 101 is doing is a different approach. It is a transposon-based approach, a different technique to getting the T cells transfected with this T cell receptor. And they feel that they may be able to get a more earlier or more naive immune cells to be transfected, potentially leading to long-lived immunity against the tumor. So that is what is being, there are a couple of exciting things about the BCMA product compared to the ones we have been talking about so far. In addition, they also have what they call a safety switch built into it. So if the CAR-T does lead to too much of side effects, there is a potential for us to be able to turn off those CAR-T cells inside the, even after it is infused. So there is an added layer of efficacy with the PBCMA product that is not available with the other CAR-T cells. Now, the data that was presented at ASH, it had a whole bunch of different approaches in terms of additional treatments given along with the CAR-T. Like a couple of the arms had lenalidomide, one of them had rituximab, and some of them had reinfusion of the CAR-T cells. So they're looking at a whole bunch of different approaches, all of which are important questions for the field. But I think the only takeaway message right now is that it appears that the PBCMA is certainly effective. We did see some responses, including deep responses at the higher dose levels. And we did not really see a lot in terms of cytokine release syndrome or neurotoxicity, suggesting that this is, a safe product. But I think, hopefully we will get more data. These trials accrue more patients.
All right, well, thank you so much. We'll go on then to Jack. Jack, your questions, please.
Dr. Kumar, thanks so much for being available for this. It's always so much information that you're able to provide. It's pretty incredible. So this year in 2021, we'll likely see the FDA approving a couple of the different CAR-T treatments that go by the names ide-cel and cilta-cel. And yet we also know there still will be many clinical trials for different CAR-Ts. So given, if all of these are available to patients, how will you and how will the patient decide which CAR-T to shoot for?
That's a very important question, Jack. I mean, it's a good problem to have, obviously, and hopefully we'll be there by the end of the year. My preference would be that, if you can get on a clinical trial, that's always should be the default option, partly because we are continuing to improve upon these CAR-T cells, right? So we are asking questions, can we make them better? Can we figure out where to use them? So I think that would be my default option. If you cannot get on a clinical trial, which is the case in many patients because of very strict entry criteria for these clinical trials, then I think we need to consider using one of these approved CAR-T cells. But it also depends on the comfort level of the patient too, right? If they don't feel comfortable getting on a trial where we don't fully understand how effective a particular agent is, that is totally understandable. Finally, I think even with lymphoma, it's taken years to really kind of cut through all the red tapes associated with the reimbursement issues. So I suspect it's just not going to be any different for myeloma either. Even though we may have the products approved by the end of the year, it might be another year or two before we totally figure out all the funding agencies, how they're going to reimburse for these products.
Great answer. You also mentioned earlier that most of the CAR-Ts and most of the bispecifics that are in trial target the BCMA antigen. As such, if you're treated with one of these and then you possibly relapse, would you still be comfortable considering another BCMA targeting treatment? Do you have to wait for a while? Would you go to one of those CAR-Ts or BiTEs that target a different antigen? How do you advise on that?
Jack, that's going to be an important question coming up for the future as we have more of these drugs available with BCMA. So if you look at the BCMA targeted, right, there are three classes of drugs. There's the antibody, the ADC, like the belantamab. There are the CAR-T cells, and there are the bispecifics. Now, there's little in terms of actual data to say that one works after the other, but theoretically, you could argue that, and something like an ADC, which works through an entirely different mechanism, there is no reason why we can't use a T-cell based therapy after someone relapses on belantamab, because even at the time of relapse from belantamab, the myeloma cells still express the BCMA antigen. But a more difficult question would be, if we have kind of flogged the T cells already with one of these treatments, will the other one still be able to recapitulate some of these effects from the T cells? I don't know the answer to that. I think that needs to be studied. Theoretically, I think they should work because they're different approaches, but practically, I think we really need to have the data. But it may not be a problem that we may have to continue to face for too long. We already saw two different bispecifics that were present at ASH, the talquetamab and the cevostamab, both of which are targeted towards non-BCMA antigens. And I anticipate that those choices are only going to continue to expand. So we may not have to really deal with this question ever, even though it seems like the most important next question.
Good. And then lastly, although I can ask more, if a CAR T patient continues to do well clinically, I know that they are still being measured for the amount of re-engineered CAR T cells that are still being seen in that patient. If the amount of CAR T cells decreases, is that necessarily a prognosticator that the patient will in fact relapse?
Not necessarily. I mean, most of these CAR T cells, they tend to disappear by 6 months at this point in time, at least disappear to the extent that we can detect them. I don't think it means they have, patients don't have those T cells around anymore. Some were hiding. Till now, there has not been very good correlation in terms of, a defined time period between them becoming undetectable and the myeloma coming back. It's all over the spectrum. But we do know that people who don't mount that initial response, they are less likely to get a response in the first place and relapse soon.
Okay, good. Thanks so much, Dr. Kumar, and I'll hand it over to Cindy.
Thank you, Jack, and thank you, Dr. Kumar. Yeah, I had tons of questions I could ask too, but I'll stick to my 3. Maybe sneak in a 4th one. I don't know. But there was one that came to my mind when you were talking before. And I'm wondering, is there like a sweet spot as how many myeloma cells that you need to have in your bone marrow for CAR T cell to be effective? Is there like too few T cells and too few myeloma cells and the T cells won't have anything to recognize, and too many, it will be too great of cytokine release syndrome?
That is the $1 million question, considering it's about $1 million for each of these therapies. You need enough of the tumor cells to mount an antigen response, right? So I think as we're starting to move into the early lines of therapy, we don't know the answer to that question if there's a minimum number of myeloma cells that need to be there. But in the setting of other diseases like lymphoma, there it appears that even with a lower tumor burden, they do develop an adequate response. So I think I'm hoping and expecting that the data would be similar even for the myeloma setting. And it is going to be important because especially if you're going to move this in the upfront setting as a way to get rid of remaining disease after conventional therapy, we really want to find out if there's a threshold effect. And that is one thing in terms of the actual numbers. And second is we also know that the myeloma cells tend to become more and more heterogeneous as they go through relapses. Having a wide spectrum of antigens, does that have something to do with a more robust immune response compared to a diagnosis where they tend to be more homogeneous? We don't know that either.
Okay, thank you about that. And I guess CAR T cell therapy is hinged on T cells. And I've been learning that there are many different types of T cells and that they're not all the same. Is there a subset that might be better to be used for CAR T cell therapy? And also, could you address, I keep on hearing that some people may not be eligible because their CAR T or their T cells are exhausted. So how do you measure your viability? And if T cells get exhausted, should you harvest them and store them like you said, store stem cells? See, I snuck a lot of questions in one.
Yeah, no. So with respect to the T cells, you can't really, there's a minimum amount of T cells that you need to collect from the apheresis sessions. And there's clearly some individuals where we may not be able to collect adequate, there's a failure in terms of the manufacturing process because we just don't have good quality, adequate numbers of T cells. There is no real measure that we can do before we do the apheresis to see if that is going to work out or not. Typically right now, it's a question of collecting, trying to make them, and you fail at making those T cells. Now, how can we get around that, right? So I think a couple of things. One is obviously, as you mentioned, when patients get their transplant in the beginning, do we collect some T cells and store them away? That's an exciting thought. Would it be same thing for even smoldering myeloma patients? Should we collect some T cells from those high-risk smoldering myeloma and store them away for future? I don't have a right answer for that question because of, again, the logistics and the cost involved with many of these things. The second is and I don't know if you're going to be talking about the allogeneic CAR T cells, but that's another approach where you can take a normal volunteer T cells and convert them or change, modify them to be used. And we saw data at ASH from the Aiello 715, which was the first allogeneic CAR T cell to be used for myeloma. And we did see responses, and we did not see any graft versus host disease, suggesting that it is also potentially a safe and effective way to approach, and might be the way to do that for some of these patients where we are unable to make their own autologous T cells. But Cindy, I think that maybe there was one more question buried in there, which I forgot.
Yeah, I was just wondering if there's a specific type of T cell that should be used.
Right. So I think one of the approach has been to use the earlier stages of T cells, right? More of the memory phenotype cells, with the hope that those T cells will live longer and is able to provide an anti-tumor immunity over an extended period of time than what we are doing right now. So there are studies looking at that, whether it be the PBCMA1 or there are other CAR T cells also targeting that approach. So I think, it's again a theoretical possibility. We don't have the data yet to say that those approaches will lead to a more durable response.
Well, thank you. Now, my next question is, although CAR T cell therapy is amazing in the myeloma population, we don't see the long-lasting results that we see in some of the other indications. Can you talk about maybe some of the trials that are underway or studies that are underway to see ways to improve the efficacy and sustainability of CAR T cell therapy?
Absolutely, Cindy. So if we talk about the data from the BB2121, for example, we saw the median progression-free survival was just under a year. But the concern there have been that it's such a late stage. The myeloma is so rapidly growing that we just don't have enough. Even when we get them down to very low levels, they still grow back up. So different ways of getting around, making it more effective, right? One is to use it in the earlier setting. There are phase 3 trials that are looking at the time of early relapse, randomizing patients to CAR T versus the triplets like the ones we talked about earlier today. There are also other trials that are looking at high-risk patients, even as part of their initial therapy, either with a transplant or without a stem cell transplant. And those trials will give us a good sense of, can we improve the efficacy by doing it earlier? Then we are also trying to change the efficacy and durability by using these different approaches in terms of what type of T cells we include in that population. I mean, there's some suggestion that early approach is better, more durable, because there was data from the Chinese studies where the patients were not that extensively pre-treated, where you have the progression-free survival that is in excess of 2 years. So it seems reasonable to assume that will be the case as we move in the earlier.
How about dual targets? Is that something that may increase efficacy?
What is that? Sorry.
Dual targets, having two
dual
targets.
target, yes. So there are, sorry, we didn't get into that. So there are two different approaches. Obviously, we can make CAR T cells that could do one or more of the antigens against, or else you can also use sequential T cells, CAR T cells, and that has been done with a couple of studies, one from China where they did a BCMA as well as a CD19 based CAR T cells to see if that would increase the efficacy. And the hypothesis there is that maybe some of those myeloma stem cells, so to speak, or early myeloma cells might be CD19 positive, and so by getting rid of the more mature tumor cells and the more immature tumor cells, we can get a more durable response. Whether that pans out, I guess we'll have to wait and see.
Right. Thank you so much.
All
Gary,
right,
I won't
thanks.
ask any more at this point.
Yeah. No, well, your third one was actually a 4-part question, Cindy.
So
In
thank
any
you.
event, yeah, Priya, any questions
Yeah.
from the audiences?
Yeah, we have a couple of questions. Dr. Kumar, what next? CAR T cell therapy does not work.
What if the CAR T cell therapy does not work?
Yes.
So what do you do next?
What do you
Sure.
need next?
Well, the good thing is we have so many other options, right, for other treatments. So let's say if the CAR T cell either didn't work or worked and then stopped working, we can certainly look at other treatments we have, the more traditional types of treatments. There is always the bispecifics that are coming along. And one of the very interesting finding from the presentation from the BB2121 at ASH was that even though the progression-free survival was under a year, the overall survival for that cohort of patients was about 3 years. Now, one could argue that this is because it was a highly selected group of patients because of the clinical trial eligibility, but it also brings up the question that do we kind of reset the disease to some extent by making those myeloma cells down to that MRD negative stage before it grows back up? Or do they become more responsive to the treatments that they were not responding to before? I think that is an interesting concept. I think we just need a larger body of data to say that's true.
Thank you, Dr. Kumar. Next question is, will CAR T cell therapy replace stem cell transplant in the future?
Well, I don't, I think it might. I don't see that happening in the next 4 to 5 years because of the extent of the studies that needs to be done. But I think the proof of concept is going to be in the high-risk patient population where it's being studied, right? So I think especially in those populations, if we can show that the CAR T cell actually are giving you more durable responses than what is currently achievable with the stem cell transplant, I think that sets the stage for definitely displacing the transplant from that point of care. Whether we will stop using transplant completely for myeloma ever, I don't think it's that happening in the next 10 years or so, because until we have a cure for the disease, I don't think we're going to be throwing anything out the window.
Thank you, Dr. Kumar. One more question. Can you speak specifically to relapsed and refractory myeloma patients who have undergone many lines of treatment and how CAR T will or won't be an option for them?
So I think for the people who have had multiple lines of therapy, I think those are the ones who would definitely have access to the CAR T when it gets approved, because again, we don't know what the final label would look like, but I suspect it's going to be for patients who have exhausted the currently approved treatments. So yeah, I think for those patients, CAR T would be the appropriate approach, or any of the immunotherapies.
Thank you, Dr. Kumar. I think we've been discussing in detail about CAR T cell therapy in myeloma. So thank you so much for all the information shared. Gary, Jack, and Cindy, thanks for joining today. This talk and transcript will be available on curetalks.com as well as on our YouTube channel. So stay tuned for more discussions on curetalks.com. Thank you, everyone. Have a nice day and stay safe.
Thank you.
Thank you.